in this wonderful building, the Clark Center. This is the home of Stanford's Bio-X. I like to think of the Clark Center as a kind of Noah's Ark, in the sense that this is a building that houses two from every discipline that's relevant for the mission of Bio-X. Initially, the idea was to bring all these people together and encourage them to interact with each other, to embark on new kinds of experiments, to tackle and understand life systems complexities, particularly as they pertain to the human body and health and disease. We have all of these disciplines mixed together in this wonderful arc. And over the years, what we've discovered is that these collaborations have resulted in really extraordinary discoveries, bringing together engineering approaches to work on, for example, muscle contraction, or bringing neuroscience together with electrical engineering to work on brain-machine interface, or bringing math, computer science, and statistics to work on understanding our own personal human genomes. The whole area of high-throughput DNA sequencing, which takes advantage of amazing new microfluidic devices, is a really exciting success story. And one really important aspect of this particular success story is the idea that Bio-X is also providing what we call innovation labs.
Well, I'd say we're gizmologists here. That is, we try to develop new biomedical technologies. We start with clinical needs. We try to understand those needs very well. And then we invent new approaches for solving the needs. One of the most unusual features of our little space here within the Clark Center is what we call the Biodesign Collaboratory. It's where we make stuff. It's a place where you go early with your idea to make a rough prototype and to actually see whether that thing can work. It's part of the inventing process. Innovation is discovery plus implementation. It's discoveries put to use. And we take seriously the putting to use part of it. They have to be implemented in order to be real. For example, the fellows from a couple of years ago saw that in India, in ambulances, there was a splint being deployed for broken legs that cost $120. And the ambulance crews only could afford one of these splints, and so they get the patient to the hospital parking lot and take the splint back because it was just too expensive. So they, fast forward, they invented a pressed paper technology for a splint. Works really well, costs less than $5. These students coming out of our program have started 26 companies, and we're proud of the fact that technologies invented here have already been approved by the FDA, used in this country, used around the world. We've treated 150,000 patients already with technologies invented here.
And this idea that what's invented at Stanford doesn't stay at Stanford, but it actually is really disseminated and made available quickly, really has also been translated into other innovation labs. And a wonderful example of a newer lab is the Optogenetics Innovation Lab, an invention that allows you to control the activity and behavior of cells in your body with light. A wonderful new invention. Everybody wants to use it. All of these things have been happening in the Clark Center over the last 10 years and have produced quite marvelous results. To me, one of the most exciting aspects of Bio-X is our ability to recognize emerging new ideas at the frontiers of different disciplines and try to jump on them and help faculty and students really move quickly ahead to develop new disciplines and to develop new technologies. And one of the areas that's most exciting at the moment is the area of neuroscience. Bio-X Neuroventures, in collaboration with faculty really across the entire university, including many of our clinical colleagues, has helped now to establish new brain initiatives at Stanford. And in a real Bio-X-like spirit, this initiative covers the entire campus, all the way from clinical neurosciences, neurology, neurosurgery, psychiatry, all the way to the business school and even the School of Education. So in my lab, we want to know what's going on during critical periods of learning in the kid's brain. And we want to understand the molecules and mechanisms that are responsible for allowing for these amazing periods of rapid learning, huge, robust learning, and what brings those periods to a close. Wouldn't it be great if someday we could take a pill as adults and open up those brain circuits again to the kind of early plasticity that's present during development? For example, one of the graduate students in the lab, who's a Bio-X fellow, has been working on the problem of stroke.
So the main project that I've worked on here, this collaboration with another lab at Stanford through Bio-X, was getting at this idea of, well, if you can increase plasticity in the brain by getting rid of molecules that normally negatively regulate plasticity, would you get a better outcome after some type of acute brain injury, such as stroke? It's a really exciting time to try to understand brain plasticity, or this idea of how do connections change in the brain. I think there's a lot of excitement surrounding this question. If we can manipulate circuits in the brain, can we get better recovery later in life? Can you enhance learning perhaps later in life? And tying that together then with this really compelling first question of how do we get this really complex, wiring of the brain in the first place?
The culture at Stanford is one reason I think that Bio-X and the Clark Center is so successful. Colleagues here, faculty and students, are really thrilled to collaborate and to interact with each other. And I don't think it's an accident that Stanford's Bio-X program has grown up here in Silicon Valley, which is another place that many people understand the value of both high-risk, high-payoff approaches to invention.
We're in an uncharted territory for the most part, areas that don't fit into any one scientific discipline. The environment of having engineers, physicists, chemists, biologists, along with physicians, facilitates out-of-box thinking so that we can tackle our problem of interest, early cancer detection, in unique ways that otherwise would not be possible. And the Bio-X fellows are a perfect example of this. These fellows are the people that bring those fields together by being the glue between laboratories that otherwise wouldn't talk to each other. And I think that's an example of what makes the Bio-X Fellowship very successful. The focus of our work, the Gambier Laboratory, is predominantly cancer-related research. We're specifically looking at ways to detect cancer early. These include techniques in molecular imaging, how we can identify small areas of tumor that may be somewhere hidden in your body. The technologies that are unique to the lab include new kinds of molecules, so molecules that are basically spies that are injected into your body. There are things that we're working on in the lab now that we started 12 to 15 years ago that are only now making it into the hospital setting. It means that you have to have people that are willing to believe in things for the long haul, to take risks, to invest in high-risk, high-return research, but more importantly, to have the patience to stick with it.
What's really amazing about the Clark Center, and it's something really special to Stanford, is it is located physically right at the intersection of all of the disciplines that are represented in the Clark Center. The medical school is right over here, and the School of Engineering is right over there, and biology and chemistry and physics are right over here.
And the hub of all of that is right here in the Clark Center, right? We're at the crossroads where it's easy for people to come over. This place is magnetized to bring people in. Perfect for what we do, where we have to have a mix of these different flavors of young innovators.
I think that's played out over the last 10 years. There's a lot of people that have come into the building, others that were already in the building that, because of the physical layout, probably collaborate more. But the problem is they never want to leave. And so it becomes a Hotel California. You can check in, but you just can't check out. You could check out, but you don't want to.
It's amazing that this building actually houses 45 research labs and over 800 people working in those labs. There are over 600 members of Bio-X distributed all across campus. There are really, literally thousands of people who have key access to our buildings here and are welcome to come and use the resources and interact with people in the building. Many of the events that we hold in the Clark Center, including symposia and talks from students and the poster sessions, are all designed to really share that knowledge hot off the press before things are even published. It's amazing. The kinds of interactions, the things that happen at Stanford in this building and in Bio-X are so mind-boggling. And what I especially like is being able to empower the great minds of Stanford faculty and students to allow them to do what they are really best at, which is invent and discover and disseminate.